Bismillahirrahmanirrahim. We start A-level biology 9700. This is a paper 5 which has been solved and explained, and the techniques for doing paper 5 will be discussed in this video. This is the June 18 paper 5-3, and we will start discussion on it, how to read the question, how to attempt the paper. As I often tell you, the paper 5-3 is on the same day as paper 2, and it's for 1 hour 15 minutes. And paper 2 is for 60 marks, and that is also for 1 hour 15 minutes, and this is also for 1 hour 15 minutes, but this is only for 30 marks. So you need to think, why is Cambridge giving you so much time for a 30-mark question? The reason is because it's a paper in which you need to think, you need to plan, you need to then write down the answers. You don't need to immediately just read the question and write the answers. You can probably finish the paper in half an hour. But why have you been given so much time? Why have you been given 1 hour 15 minutes? Is for you to really not write down the answers immediately, but to think about each question and then think that you're doing this in the lab, and how would you be executing it in the lab, or how would you be doing it as an experiment. So that is what you need to understand, not just start writing immediately and finish the paper in half an hour and walk out of the exam hall. Now we start doing question 1. Sodium hydrogen carbonate indicator solution contains sodium hydrogen carbonate. Makes sense? Some students found out on the internet that this indicator solution changes color with pH. The students also discovered that this indicator is used to determine the concentration of carbon dioxide in a solution. The students decided to use this indicator to measure the rate of photosynthesis, in algal balls. Algal balls are unicellular algae. Algae are unicellular plants that are immobilized in alginate. The student assumed that the rate of carbon dioxide uptake was proportional to the rate of photosynthesis. The rate of carbon dioxide uptake was proportional to the rate of photosynthesis. Figure 1.1 shows the apparatus the students used. Now let's look at the apparatus and think we are doing this in the lab. So as you look at the apparatus, it's got a lamp. Now we look at this lamp, and then we've got a glass container, and we've got an indicator, and we've got the algal balls, and then we've got a ruler, and at the moment this is now here, this is the lamp, and this is 25 centimeter. So look at it very carefully. Glass container, indicator. I always tell you a lot of things is, number one, if there's any fluid that we are experimenting here, then it must have the same volume must be used, and the same concentration must be used. Same volume and same concentration. Like if I say a 5% sucrose solution, or a 5% hydrogen carbonate solution, or a 5% glucose solution. So volume, are you going to specify the volume? Is it 20 ml? Is it 50 ml? Is it 30 ml? So volume, concentration. Whenever is the fluid in a paper 5, you must specify the volume and the concentration, or you must keep it constant. A laboratory technician prepared a series of test tubes containing hydrogen carbonate indicator solution from pH 7.6 to pH 9.2. A laboratory technician prepared a series of test tubes, 1, 2, 3, 4, containing hydrogen carbonate indicator solution from pH 7.6 to 9.2, 7.6 to 9.2. The color of the indicator solution in each test tube is described in table 1.1. Now let's look at, so this one is light yellow, dark yellow, then light orange, then dark orange, then red, then light magenta, dark magenta, and then purple, light purple, and then dark purple. Now, on this side, we have increasing carbon dioxide concentration. On this side, we have increasing carbon dioxide concentration. So that means here there will be more carbon dioxide. And on this side, we have decreasing carbon dioxide concentration, so less carbon dioxide. Now you have to understand, if we're going to study photosynthesis, photosynthesis takes up carbon dioxide. So if there is more photosynthesis, it will be towards this side, less carbon dioxide. And if there is less photosynthesis, then there will be more carbon dioxide. So you have to correlate the carbon dioxide concentration with the photosynthesis. You see, photosynthesis is what? CO2 plus H2O gives you glucose, C6H12O6. So more the glucose formed, more the CO2 will be taken up. So the CO2 must decrease when there is more photosynthesis. Now part A of the question says, suggest what was added to each of the test tubes to maintain the pH. You know that a buffer solution is used to maintain pH. So a buffer solution was added, or a buffer was added. You just have to write buffer and you got your 1 mark. The B part of the question. The B part of the question says unicellular algae, you know, are unicellular plants, can be immobilized using a technique that is similar to that used to immobilize enzymes. Outline the method that the students could have used to immobilize the algae. Now it's very simple. Mixing the algae with sodium alginate, adding the alginate and algae to calcium chloride, and using a syringe or a pipette to drop to form the beads or the balls which they have mentioned. So mixing the algae with sodium alginate, this you do in paper 3. We've done this many times in A1. Adding the alginate and algae to calcium chloride, and then using a syringe or a pipette to drop this so that it sort of coagulates and forms these algal beads or these algal balls which they have mentioned. So this is to immobilize the enzyme, because now they are in this ball or this bead, and now the enzymes are going to be used to do some work. So these are called, this is an example of immobilized enzymes. Now let's look at part C of the question. The students use the apparatus shown in figure 1.1 and the colors described in table 1.1. So this is figure 1.1 and this is table 1.1. To investigate the effect of light intensity on the rate of photosynthesis. So two things are being investigate: what? The effect of light intensity on the rate of photosynthesis. The students propose the following hypothesis. As light intensity increases, the rate of photosynthesis increases. So identify the independent and the dependent variable. Now I've talked about in a previous video as well. Independent variable, I always say, is the thing that you, or you can say I for independent, is the thing that you change. Now what are you going to change? You are going to change the light intensity. You're going to vary the light intensity. How? We're going to discuss that. And what is the dependent variable? What are you going to measure? Because you're not measuring the rate of photosynthesis. You're going to measure the color of the indicator or the pH of the indicator. How the pH is going to change. Is it going to change towards this side, or it is going to change to this side? Is it going to change to this side? Are we going to have less carbon dioxide, or are we going to have more carbon dioxide? So is the pH going to change to 7.6, or is the pH going to change to 9.2? So this is what you have to investigate. You're going to look at the pH. So then that is why the answer to this part is color of the indicator, not rate of photosynthesis. You do not write rate of photosynthesis, because rate of photosynthesis can be measured by many ways. You can measure it by how much oxygen is being produced. You can measure it by how much glucose is being produced, or how much starch is being converted, the glucose being converted to starch. So rate of photosynthesis here is being not measured by that. We are measuring it by the color of the indicator, or the pH, how the pH is going to change. So this is what you have to write. You do not write rate of photosynthesis. As I tell you again and again, rate of photosynthesis can be measured in many ways. You can even calculate the volume of oxygen produced per unit time, and that will give you the rate of photosynthesis. So what are you going to be measuring? Dependent variable is what you are, what the result, what are you going to be measuring? You're going to see the color change of the pH or the pH change which they told to you in the earlier part of the question. Then we look at part 2 of the question. The part 2 of the question says, describe a method the students could use to collect the data needed to test their hypothesis. Your method should be set out in a logical way and be detailed enough to let another person follow it. You should not repeat any details from B about how to prepare the immobilized algal balls. So the first thing is, let's talk about the independent variable. Now you only have to give me 5 points out of all these which are given. First of all, you have to give me how are you going to vary the light intensity by either increasing the number of lamps or changing the distance between the tube and the lamp. And then you give me 5 distances, and the 5 distance could be anything between 10 and 200 centimeter because they've given you 25 centimeter in the diagram. Then cover the tubes with metal foil, then expose to light and record the color change. The point that expose to light and record the color changes, because if you do not cover them up and then suddenly expose them to light, well, that is the point that you start timing and you sort of look at the color change, how much time it took for it to become purple or become the other side if it went. So control, and how would you use a control? The control is very important that you talk about. Control is, as I tell you always, control is that whatever is the enzymes, that thing should be either boiled or you should remove that. So boiled algae or no algae, that would be the control. Then as I told you about the fluid part, same volume or same concentration of the hydrogen carbonate solution. So this is what we were going to talk about, same volume and same concentration. You could say take 20 ml or 30 ml of 5% hydrogen carbonate solution. That would get you that mark, and that is what you keep constant. Then the same number of algal balls. So same number of algal balls. You could also say the same mass. There are a number of things which are allowed in the mark scheme. Then this is something which you can write in every designing. Repeat. Repeat 3 times to remove anomalies. Anomalies means the ones which are the errors. Like for instance, you're getting a reading of say 10, 12, and then 11, and then suddenly you get a reading of 30. Well, that is not, there's something wrong with that reading because it doesn't fit the trend. Then you talk about the safety part of it. So this is a medium risk experiment. Why? Because here you're only handling 2 things. Number one, algae, which of course some people could have allergy to it. Like I'm allergic to mangoes. If I touch mangoes, I get a rash. So you use gloves, allergy to algae, or the chemical, the sodium hydrogenate carbonate solution is harmful. So you use gloves and you wear goggles so that God forbid it spills, it doesn't fall onto your eyes, or the algae could cause some sort of a rash on your hands and you touch your face or your eyes and you could get some allergy to it. Now this was only for 5 marks. So any 5 points, but then of course it's important that you give me the 5 points which are very relevant to this practical. Then let's look at the part 3 of the question. The part 3 of the question was very clear that the students were concerned that the lamp may have increased the temperature. Naturally, if you have a light source like a bulb, it also gives off heat. But it's also giving you light, but it's also giving, if you put your hand against the bulb, there's a little bit of heat which is also being generated. So lamp may have increased the temperature of the solution in the container. Suggest how they could have attempted to control this variable. Place a heat filter or a heat shield between the lamp and the container, something which is going to absorb the heat but will allow light to pass through, something which will allow a heat shield is just 2 pieces of glass with water in it. So it allows light to pass, but it won't allow the temperature to change. So this is what you have to say, a heat filter or a heat shield. Then let's look at the D part of the question. The D part of the question says, the light intensity can be calculated using the formula 1 over distance whole square. So using the formula in figure 1.2 to calculate the light intensity for the experiment shown in, and this is what we had. We had this 1.6 into 10 raised to the minus 3 or 0.016 or 0.02. 0.016 or 0.0016 or 0.002. So this was the answer, and this was a 1 mark for you to calculate this. The student decided that using a pH probe would give them more accurate data. They decided to measure the pH change for one value of light intensity. Figure 1.3 shows their results. So we've got pH here on the y-axis, and here we've got time in minutes. So you see this graph, which you can see is here. This graph is increasing like this, and then it becomes constant, a very typical graph. and light intensity is being kept constant. And here we're seeing the graph and how the pH is changing, and then the pH remains the same around about 9.2. Now it says state why the use of a pH probe provides more accurate data. Well, pH probe is more sensitive. It gives you a digital reading. It's not measuring colors. Colors can be very variable. Somebody can see one color. Somebody might say differ with the other in the opinion. Well, it's not as usual light, or it's a bit dark, or something like that. That's a very variable. It's a very qualitative assessment. Here, it's a quantitative assessment. So it's much more sensitive. The data shows in figure 1.3 for a high light intensity. On figure 1.3, sketch the curve that you would expect for a low light intensity. Now I've sketched the curve in blue here, and so it will be to the right of the curve, and it will be from 0 to 10 minutes, and this is what would get you your 1 mark for that curve, which I have drawn in blue on the graph. So this is the one which I've drawn on the graph, and you can see this curve. Then it says part 3, state why carbon dioxide is not a limiting factor in this investigation. The reason is the indicator solution provides carbon dioxide, or you can say hydrogen carbonate ions, and that was, of course, was not going to be a limiting factor because there was abundance of that. In part F of the question, certain weed killers work by reducing the rate of photosynthesis. Now this is something totally different from the previous part of the question. The student modified their method to investigate the effect of 3 different weed killers on algae at the same light intensity. Modified their method to investigate the effect of 3 different weed killers on algae at the same light intensity. The investigation was replicated a number of times, and the pH was recorded after 30 minutes. The student calculated the mean pH for each treatment as shown in table 1.2. Table 1.2. The students carried out t-tests to compare the mean pH for each of the treatments that used weed killers with the treatment that used no weed killers. So we have treatment 1 in which there were no weed killers. The student calculated the value of T for each of the tests. Table 1.2 shows the results from their investigation. Let's look at table 1.2. We have treatment 1, 2, 3, and 4. Now you know it is said here treatment 1 was no weed killer. So there was no weed killer in treatment 1. In second, there was weed killer A, then B, and then C. So there were 3 different weed killers. And mean pH after 30 minutes was 9.2 to 8.5, 9.0. So this was no weed killer was 9.2. And then the one A was 8.5, and then it was 9.0, and then it was 8.7. So 8.5, 9.0, 8.7, and no weed killer was 9.2. Then the probability values was less than 0.001, more than 0.05, and less than 0.05. Now suggest a null hypothesis. Now I've told you this many times. Suggest a null hypothesis means you have to negate it. So there is no significant treatment between pH for treatment 1 and 2. So there is null hypothesis comparing the effect of treatment 2 with treatment 1. Treatment 1 was the sort of the one which was the control sort of a thing in which no weed killer was used. There was only distilled water. So there is no significant difference. Now please understand that some words have been underlined by me, and in the mark scheme there is no significant difference between pH for treatment 1 and treatment 2. And this got you your 1 mark. So nobody should be missing out this 1 mark. It's very absolute that you get this 1 mark. We come to part 2 of the question. The part 2 of the question was the t-tests were used to compare the means. We usually do this to compare the means. You must revise t-test. State one feature of the data that allows use of the t-test. Continuous data is normally distributed, size of the sample is less than 30. So these are the 3 factors in which we use the t-test, and these are the factors we must know why and when do you use the t-test. When calculating the effect of weed killer A on the rate of photosynthesis, 15 degrees of freedom was chosen. The students carried out 7 replicates of treatment 1. So state how many replicates were carried out for treatment 2. Now part 3 of it can be understood in a very simple manner. When calculating the effect on the rate 15 degrees of freedom. So what I always say is you add 2 to it. So this is 17. 15 plus 2 is 17. The student carried out 7 replicates. So how many repeats were carried for student 2? It has to be 10. Why? Because whenever we calculate the degree of freedom, it is n1 minus 1 plus n2 minus 1. So you need to add 2 to the degree of freedom. So the degree of freedom, it says, was 15. So 15 plus 2 is 17. Then we are told that 7 was, this was 7 was calculated. So it has to be 10 here. So 10 plus 7 would be 17. Please understand this. They have given you that 15 degrees of freedom was used. So 15 plus 2 is this plus 2. And then we had 7. So 15 plus 2 is 17. 17, so 10, 17 minus 7 here. So 10 is left. So this is n is equal to 10. Then let's look at the last part of the question, which is the first question that finishes your 22 marks of questions of the paper. This is a 30-mark paper. The students concluded that the weed killers had a significant effect on the rate of photosynthesis. Explain to what extent the p-values, the probability values in table 1.2 support their conclusion. Yes for weed killers A and B. A and C, because p-value is less than 5%, results are due to chance. And effect of weed killer A greater than weed killer C, because p-value is greater than 0.05. Now you need to understand this. What is the probability value, and when do we look at it when it's less, and when do we look at it when it's more, and what does all that signify? The second question was on lionfish are a type of fish native to the tropical waters of the Indian Ocean and Pacific Ocean. Lionfish were first reported off the southeast coast of USA in the 1980s. Lionfish are an alien species to this area. Their lack of a natural predator in these waters resulted in their numbers increasing dramatically, allowing them to migrate to new areas. They have now become established across Caribbean. Their numbers are so high that they are now a threat to many native species. In 1 year, it was estimated that 5 million prey were consumed by 1,000 lionfish. Express these data as a ratio. So the ratio has been given, ratio 1 has been given here. And what you had to figure this out is what was the ratio, what was it to 1. So this is 5,000 to 1 was the ratio. Then coming to question B, part B of this question, surveys to assess the changes in lionfish abundance have been carried out. Every year since 2004, divers have carried out visual transects at 9 sites along a 15-kilometer stretch of continuous reef off the coast of one island in the Caribbean. Visual transects involve swimming in a fixed direction across the reef for a set distance and counting the number of lionfish. State one variable which was standardized in these surveys. Length of the transect, because it said fixed direction at a set distance and counting the number of lionfish. 9 sites along a 15-kilometer stretch of continuous reef. So length of the transect, number of sites surveyed, geographical area in which the study was carried out, and the direction of the sampling. So you have to just pick up something which was given in the question. Now that is why I say you have to read the question very carefully in order to pick these points up. The mean abundance of lionfish on the reef was calculated for each year. These values are shown in figure 2.1 using a log scale, and the bars show 95% confidence intervals. Mean abundance of lionfish in the log scale, and these are the years that we are talking about. So we need to be looking at this. A group of students read an article stating that local people were being encouraged to catch and eat lionfish in an attempt to reduce their numbers. The students concluded that local people started catching and eating lionfish in 2008. Explain to what extent the data in figure 2.1 supports their conclusion. Look at the supports. Lionfish abundance has decreased. Not support increase in mean abundance decreasing from 2007 to 2008. So could have started catching lionfish earlier in 2007. And then 95% confidence interval bars overlap. Therefore, no significant difference between means. Coming to the C part of the question, some local communities hold annual fishing trips where they are encouraged to catch as many lionfish as possible. In 2010 and 2011, 16 boats took part in these fishing trips. The mean number of lionfish per boat and 95% confidence intervals were calculated. Figure 2.2 shows the result. Now you see the mean number of lionfish and you see per boat and you see the year of the catch. Then it says the 95% confidence intervals are calculated from 2 into SM. Standard deviation can be calculated by rearranging the equation for standard error. So standard error, standard deviation, number of boats. You know the number of boats was 16. And the standard error here, the SM was 2 into SM was 35. This is what you had to see here, which I've just shown you where we got this 35 from. Last part of the question is use the data in figure 2.2 and the equation in figure 2.3 to calculate the standard deviation of the mean number of lionfish caught per boat in 2010. And from the graph, 95% confidence interval is 35, which I've just shown you here, how we got the 35. And then we've got this here. So SM is equal to 35 over 2 is equal to 17.5 and under root n is equal to 4. You know, n was 16. And then you get this answer, and this is 70. If you got 70, but you got your 3 marks. So this is the math that you were expected to know, and that finishes your paper 5. Please go through the parts which are not clear to you and read those through the book again, and then figure out where you missed out which point and where you got this wrong. So all the best, and I will following up with another paper 5 very soon. And thank you very much.